Hello and welcome to Biology 215. This is my overlap of the lecture. Basically, I'm lecturing out of the book, and what I talk about is in the book, and what is in the book is kind of what I talk about. One of the most important things for this particular class, though, is that you know and understand the things that I do highlight in my lectures. Today, hopefully, I'm going to talk a little bit about what life is. I'm going to talk about the classification of life. We're going to talk about biology and about what is science, and we'll talk a little bit about how we do science. One of the things that I really want you to be able to do by the end of this class is to be able to do and evaluate science. So we have a number of different ways that we kind of define life, and these look at structure and these look at the function of what is life. So life is a number of different things, and the main thing that your book talks about life is this ability to have order, evolutionary adaptation, regulation, homeostasis, reproduction, response to the environment, growth and development, and energy processing. And so anything that we look that's alive today is going to basically have all of these 7 characteristics. So what you want to be able to do is to be able to know and describe these characteristics, and anything that I would give you that you'd be able to tell me this is alive, this is not alive, be able to talk about those 7 characteristics with that. Again, the cell theory is a big thing that in this class what we're going to say the smallest unit of life is going to be a cell, and that you need to know and understand kind of what cells are. The second thing that we're going to look at in this class is basically the levels of life. We look at organization, and we look at kind of arrangement. The smallest things that we are going to be looking at are basically cells, although we will look at some of the processes of cells. And that we see is if we group these together, we group cells together, we become tissues. If we group tissues together, we become organs, organ systems, and populations. And again, we will be looking at these levels of how things interact with one another. The first organisms that we're going to be looking at are kind of basically single-celled organisms, and we're going to be looking at how they interact with one another. And again, we will be looking at how ecosystems interact with one another, specifically our ecosystems. One of the things that you need to be aware of is what emergent properties are, is that basically emergent properties are those properties that result from the arrangement and interaction of parts within a system. And if you want to learn more about that, you can go ahead and talk about to that with me. Organisms interact with their environment in a number of big ways, and again, in ecology, we will look at this a little bit, but basically energy moves into an environment. Energy moves into an environment in terms of sunlight, and it leaves the environment in terms of heat. Elements, matter, like carbon, kind of remain in an environment, and so the carbon in a tree gets converted into carbon dioxide by these herbivores, which then trees can use again to their meter. So there is a system here, and we will talk about this chemical and nutrient cycling in this class quite a bit. A core theme is basically evolution, and what I wanted to do is to kind of explain to you what living organisms that are out there, and then we'll talk about how these organisms have evolved, kind of rather than your book kind of goes the opposite way. It first talks about the mechanism, evolution, and then it talks about these organisms. And so what we're going to say is that evolution, or what we call descent with modification, or change in allele frequency over time, is basically this theme that basically describes, and everything in biology responds to this. We will look at and classify the diverse thing that we basically call life in this class. And again, there have been over 1.8 million species recorded on this planet so far. The basic idea for grouping species is to basically put things that are alike to one another. An idea is to categorize things by their inherited relatedness, okay, evolutionary relatedness, just how they look at one another. And again, we did this, if we look at this, we have a domain which is the largest group that includes things that are similar to one another. And this domain here is looking at eukarya. Again, in the domain we have subgroups. I'm not going to have you memorize these, but if you look at this, these groups have less and less individuals as these individuals are more and more related. So again, if you look in this genus right here, we have these individuals in the genus, and they look more similar to one another than, again, that individual would be in there. So these are, again, in this grouping, they are like one another's, but they're not over here, and we will talk a little bit more about those groupings. The largest groupings that we should talk about are the eukaryota, the eukaryotes, the archaea, and the archaebacteria. And again, we will hypothesize some of the relationships between these, but basically they are equally as important to one another. Another thing that you need to be aware of is the different types of cells. So when we look at cells, and this isn't a domain or a grouping, we have what's called the prokaryotic cell and the eukaryotic cells. Eukaryotic cells as a whole tend to be a little bit larger, and they have more processing. They can do more. Prokaryotic cells, on the other hand, are a little bit smaller and don't do as much. The example that I use of that is basically a calculator. Your phone can do a calculator, but again, sometimes when you just need to add a number and the calculator's right there, it's a little bit easier to use the calculator than it is the phone. And so that's basically how these guys work here a little bit. Prokaryotic cells tend to be smaller and simple. Eukaryotic cells tend to do a little bit more, and they are a little bit more complex. So again, be aware of those differences, and you can go on and look at them a little bit more. Again, these things, these two domains we call the prokarya, basically because in the past when we were first looking at these through microscopes, we only saw their structure, and hence we thought they were the same because they look the same. They are not. They are very different, and we'll look at this. And so what does this mean is that basically the domain eukarya is seems to be a lot more complex because, again, the main thing that we use to measure what does it look like, we can perceive a larger difference in the eukarya domain than we do in these other domains. And again, as we're getting tools and able to look at a little bit more, we see that that is not necessarily the case. It's actually an artificial designation in there. And so we go through this a little bit, and then changing it, classifying life, be aware of the classifications of life. The next thing that we want to do is kind of basically look at how we do science. And one of the big things that we're going to do in this class is basically observation. We're going to do some experiments too, but we want you to observe directly to serve and get information. So in discovery science, you're using verificational observations and your measurements, and this is kind of what we call data. And so you look at something, and again, when you look at something and certain behaviors occur, or something occurs, it always occurs the same. If you look at something that's different all the time, then there's something going on that you're not observing there. And so what should happen in observations, that you give somebody an antibiotic, it kills the bacteria, they get better. Okay, that's an observation. In hypothesis-driven science, which is again what we do, we actually have a formal process that we call a scientific method, and we'll cover the scientific method in a little bit. But basically in all of these we have a hypothesis. We have a tentative answer to a question, and then we test this hypothesis to see if it is true or not. Again, just because a hypothesis is shown to occur, that it's true, doesn't necessarily mean that it's a right answer. And so, again, it just means that you haven't proved it false yet. And so here's one of those troubles of that. We can try and prove these wrong. We can do a falsification, in which case we know our hypothesis isn't true. But again, until we do that, we see that. Again, observation. Scientists make observations, and then they try and test these answers that we got that depend on those observations. Again, inquiry is the search for information and and explanations of natural phenomena. We are a very inquisitive species, and so that we are trying to do this. Again, so what are we going to do? Making observations, recording data. Two types of observations that you need to be aware of are what we call qualitative data. Let's go back. Qualitative data would take the forms of recorded descriptions, and quantitative data, which is basically numerical measurements. And so if you say an organism turned right and proceeded forward at 5 meters per second, is different than the thing moved, which would be, that's qualitative and quantitative. And again, we will be talking about this a lot in our laboratory. Here, we talked about this in the laboratory. Hypothesis-based inquiry can happen anywhere. Again, doctors and mechanics and a lot of other people do it as well. But we see this individual here is making an observation on her flashlight, and she has two possible questions that she does, and she solves this and answers it. Again, what a scientist is going to do is they don't necessarily just need to solve the problem. They really want to answer the question, and that's what a scientist would do. Again, some examples of this that have occurred in the past is basically, you may have all heard that vaccines cause autisms. Again, the rates of autism diagnosis were increasing. It's increasing in a population that uses the vaccines, so the idea was that vaccines do this. Again, a number of hypotheses. Vaccine use has been linked with autism. Again, here, vaccines have not been in it. What else? If vaccines are not linked to autism, then vaccinated children and unvaccinated children should have the same autism rates. Predictions: vaccinated children should have higher rates of autism than unvaccinated children, if this is the case. And so here we have two kind of opposite predictions, and we see that basically that is the case. And so there's a number of different things that we can look at here. And so no matter how we look at this, I mean, it would be a great explanation of it, but again, that's the data is just basically not there. Again, a lot of times in experiments we do what's called a control experiment. We have a control group, which allows the comparison of the experimental group. A control group is a group that you know the results of what to expect. And so, again, if you put your if you put water into the refrigerator and expect it to freeze, if it doesn't freeze, then that's kind of not a bad one. But again, it's to test for other things. And so you have a hypothesis that when you put water in a refrigerator, that the refrigerator is cold enough for it to freeze. Now, if there's something wrong with the refrigerator and you don't get water to freeze, then you may say that, hey, the refrigeration does not cause water to freeze, but that's not the case. Again, we nest and use negative controls and positive controls. The example that I used in class is an elephant repellent. Okay, so if we just say that my elephant repellent works great, that's not possible because we did not use a control. We need to test, do an experimental and test. Again, the scientific method forms over itself a lot, and gone to this experiment hypothesis, observation hypothesis experimentation is not necessarily the way that it goes, and it's a little bit more of a circle, and we will discuss that kind of in class. Again, your book does a really good job at explaining it. So what we call scientific inquiry looks at coloration in mouse populations, and this kind of experiment has been done many times. And so basically you have two populations of mice. It's the same genus and species, and so that means they can mate with one another, but they have a different form. The mice that live on the beach are more white in color. The mice that live more inland have this more kind of a mottled look. And again, one of the hypotheses was that in the beach habitat, because a lot of the predators tend to be more visual, non-camouflaged units would get attacked a lot more than once. So here's the percentage of attacked models. So what they did is they put these models out and they see how many times. And so those models do look actually pretty good. And as you can see, the white mouse is a lot harder to see than the darker mouse. on the beach, whereas the darker mouse on the inland area is a lot harder to see than the other ones. And so, again, they've done a number of experiments on that and basically show it. Where does this all get us? The goal of science is to understand natural phenomena. We want to know. We're curious and we want to know. The goal of technology is to use scientific knowledge for some specific purpose. And again, in our purpose that I'm saying I'm using is I want to make something. I want to make some money. I want to do something. I want to become famous, etc. That's why I want to use my science. Again, science and technology are interdependent. You kind of need one in order to do the other. And so, again, to ask some of the questions that we have, we need to have technology that didn't happen a while ago. And so, again, new technology drives new science, then new science will drive new technology. Again, biology is marked by discoveries, while technology is marked by inventions. The combination of science and technology has dramatic effects on society, and we talked a little bit about that. My alarm is going off. And so we do talk about a theory, which are explanations supported by all available evidence. We'll talk about the difference between the theory of evolution and the cell theory. The theory of evolution, some people are questioning the cell theory. No one questions. So it's kind of interesting that happens. Again, what I'll leave last is to kind of look at all this diversity. Why do we want this diversity? What are we looking for in this diversity? And again, in class, I talked about a possible cancer cure. And there's a bunch of different reasons why we would want this diversity that's in here. Okay.